Good morning, everyone. I'm Lakshmi A. Menon from S7EV. Today, I'm here to present my seminar on the topic Application of Machine Learning Algorithm for Neonatal Jaundice Detection, which is guided by Dr. Surya D. from Department of EAB. These are the contents. Let's move on to the abstract of the seminar. Hyperbilirubinemia, mainly characterized by the elevated levels of bilirubin in neonates, can lead to jaundice, and if it is still untreated, it can lead to several other neurological damages. So, traditional methods of bilirubin detection involves invasive blood sampling, which can be distressing for newborns as well as their parents. This is a distressing action in babies, and it causes discomfort in babies as well as the parents. So it also leads to delays in diagnosis. My seminar covers a non-invasive method that uses image processing technique to estimate the neonates bilirubin level of bilirubin for the detection of jaundice. Mainly, this method of non-invasive method of bilirubin detection is done through capturing images of neonate skin and applying various machine learning algorithms. And this method quantifies the intensity of yellow color pigmentation, which correlates with the bilirubin levels. The proposed method utilizes color space transformation, feature extraction, and several machine learning algorithms to accurately assess bilirubin concentration. Next is a basic introduction about jaundice or hyperbilirubinemia. Jaundice or hyperbilirubinemia refers to the yellowish discoloration which mainly occurs to the baby's skin or in the whites of the eye. This condition in newborn is mainly due to the inefficiency of their liver or the immaturity of their liver. According to the worldwide statistics, 80% of preterm infants and 60% of term newborns and also the 10% of breastfed newborns were prone to neonatal jaundice. Therefore, the timely detection of jaundice is necessary in order to lower the level of bilirubin and also to prevent several other complications such as kernicterus, which is a severe neurological damage. If the bilirubin level or the jaundice is untreated in babies, it can affect their brain, also its functioning. So the traditional detecting methods typically involves invasive blood tests. As I already mentioned, traditionally bilirubin was detected by blood test, which is taken from the babies, but it is a uncomfortable uh, task, and also there will be delay in diagnosis. So image processing using skin color analysis is an innovative non-invasive approach to estimate the bilirubin levels that utilize advanced algorithm to analyze skin color changes associated with varying bilirubin concentration. Next is the literature survey. And the first paper, which is written by S. Mitra and J. Rennie, which is titled as Neonatal Jaundice Etiology, Diagnosis, and Treatment. Mainly, they came to know about the neonatal jaundice and its clinical signs in newborn infants. Around 60 percentage of term babies and 80 percentage of preterm infants, and also 10 percentage of breastfed infants are jaundiced in the 1 month of age. In the next paper, Khanam, which is written by Khanam and Al Naji, which is titled as Non-Invasive and Non-Contact Automatic Jaundice Detection of Infants based on Random Forest. It mainly discusses about a automatic ROI selection method, which is a pre-processing step in the image processing technique to detect the level of bilirubin from the infant's image. Here, mainly the automatic ROI selection is done through a DLIV OpenCV face landmark detection method, which is used to extract or detect the face of the neonates from their images. And machine learning algorithm, mainly RF, that is random forest, is utilized in this paper to detect the level. In the next paper, which is neonatal jaundice detection using computer vision system. The authors have observed that this level of bilirubin from the neonate's image is mainly detected based on the skin color analysis. That is, for skin detection, mainly color space transformation is a crucial step, which acts as a pre-processing stage. And here the color space which the image which is obtained in the RGB color space is transformed to another color space, and from that the different color channels are utilized for jaundice detection. Here they have used B and CB channels of RGB and YCBCR color space. And the next paper, real-time jaundice detection in neonates based on machine learning algorithms. Here they have utilized XGBoost algorithm for jaundice detection. Mainly, it is a gradient boosting algorithm, and it has shown a greater accuracy in estimating the level of bilirubin from the neonate's image. In the last paper, which is Engine, a dataset for normal and jaundiced newborns. This paper mainly tells about different data sets that were provided by the author to detect the level of bilirubin from the neonates images. Mainly, they have used three machine learning models, which are RF, that is random forest, XGBoost, and also KNN algorithm. Next is the objectives of my seminar. The first objective is to provide a clear understanding of what neonatal jaundice is and its causes, and also the role of bilirubin in this condition. Next objective is to present image processing technique as a modern non-invasive approach to estimate the bilirubin levels, which minimizes the need for painful blood draws. And the third objective of the seminar is to explain how skin color analysis is used to detect jaundice by identifying the correlation between skin color changes and bilirubin concentration. Next is the bilirubin level in infants. Bilirubin is a yellow pigment which is created in the body when the red blood cells present in newborns breaks down, and it is mainly because of the immature liver, they cannot metabolize this bilirubin. So this the level of bilirubin will be high in newborns. Bilirubin levels in the blood are an essential measure of liver function, and it can be linked to illness like jaundice. Newborns have higher bilirubin levels than adults because their, as I said, livers are still developing. The typical bilirubin level of an infant is less than 5 milligrams per deciliter. In neonates that were born prematurely, levels might be a little higher, which is the main cause for jaundice. High bilirubin levels that are left untreated can result in serious complications like neurological damage, and it can also lead to a condition called kernicterus, which is a condition that affects the brain when the barrier breaks down and the bilirubin enters to the brain directly to the brain and causes this condition. Next is the formation of bilirubin. This diagram mainly explains about the formation of bilirubin. Bilirubin is mainly formed by the breaking down of red blood cells present in the hemoglobin. It is a reticuloendothelial. Through the reticuloendothelial system, the red blood cells are break down into heme, and then after heme oxygenase, this heme is converted to biliverdin. The heme interacts with the macrophages, and then as I said, red blood cells will breaks down to hemo and globin. Then through heme oxygenase, this heme is converted to biliverdin, and then it is again reduced to bilirubin. This is how bilirubin is formed in infants, mainly in newborns. In the first figure, this is what I explained about the condition called kernicterus. As the level of bilirubin increases in the body of newborn, the bilirubin will enter the bloodstream, and directly it will go to the brain tissue, and then it will damages the brain function, and thus leading to the condition called kernicterus. Next is a level of risk which is a graph based on the level of risk on increasing the level of bilirubin level in neonates. Next is the existing methods of bilirubin detection. The first method is total serum bilirubin testing, which is mainly conducted through blood tests. The blood is taken from the heel, which is a painful task for the babies. And the next is urine bilirubin testing. The bilirubin can be also evaluated or detected, or it can be calculated by taking the urine from babies. And the third one is transcutaneous bilirubinometer, which is a sensor-based device. Which it has been developed recently. It can be used to detect the level of bilirubin from the patients. That means neonates or infants. But these existing methods still have some drawbacks. The first drawback is frequent blood draws. Blood is taken from the heel frequently, which causes discomfort in patients, that is newborns, and also to their parents. And next is the risk of complication. There will be many risks, such as frequent blood draws can lead to infections and several other problems in the newborns. Third is parental anxiety. This discomfort can also cause parental anxiety. And the fourth one is time consuming process, as it is already visible that the blood tests are always time consuming. And the third technique, which is transcutaneous bilirubinometer, as I said, it is a sensor based device, but it has high cost, so it is still a drawback for the existing method. Next is bilirubin detection by image processing. Image processing techniques for detecting bilirubin levels in neonates involve analyzing digital images of baby's skin to estimate the degree of jaundice. As I already mentioned, it is a non-invasive and potentially cost-effective way to monitor the levels of bilirubin in neonates. This non-invasive method mainly involves skin color analysis and utilizes machine learning algorithm to detect the bilirubin from infants' image captured using smartphones. These images are then subjected to pre-processing steps such as skin detection and region of interest extraction, focusing on areas like forehead or sclera. These are the stages in bilirubin detection. The first stage is mainly pre-processing steps or pre-processing stages, which include image capture, ROI extraction, color space transformation, and then feature extraction. The feature extractor is then given to the machine learning algorithm, which is trained by a certain data set to detect the level of bilirubin level from the given images, and then it detects whether the newborn or the infant is jaundiced or not. The first stage in bilirubin detection is image acquisition. Mainly, the image of the infant is taken by using a smartphone or any camera, and the image should have a pixel resolution of 1000, and it can be saved in JPEG file format, which can be easily manipulated, or it can be easily processed. JPEG file format or PNG file format can be used for image acquisition. Next stage is skin detection. Skin detection in the pre-processing step of image processing, particularly in mainly in bilirubin levels, involves identifying and isolating skin regions from an image. This process is crucial because the accuracy of subsequent steps like skin color analysis and feature extraction depends on correctly detecting and focusing on skin area. This skin detection is mainly accomplished by three steps, which is region of interest extraction, color space transformation, and feature extraction. These are the important steps for detecting the level of bilirubin. Uh, uh, from the skin detection, the first step is region of interest extraction. RO extraction is a critical step in image processing, which is mainly done to isolate specific areas of the image that are most indicative of bilirubin levels. The accuracy of jaundice detection is highly dependent on selecting the correct skin region of interest. So the region of interest extraction is mainly done in order to isolate the important region, that is mainly forehead or sclera, which shows a greater level of bilirubin level in infants. So the ROI typically includes skin regions that are most reflective of bilirubin concentration. Mainly in this case, we will take forehead, chest, and sclera for detecting the level of bilirubin, which shows a greater concentration of bilirubin or yellowness in their body. These areas are chosen because they exhibit, as I already mentioned, they exhibit clear color changes correlating with bilirubin levels. Infant's forehead is chosen as the ROE. ROE has been detected manually. Here we will detect ROE automatically. An automatic ROE selection automatic ROE selection is developed in this method. This ROI selection mainly consists of three phases. The first phase is face detection. Second one is face landmark detection, and third one is ROE forehead selection. So this ROE detection is mainly accomplished through a library called DLive OpenCV face landmark detection, which is a certain library which is used to extract the face from the images, and so it will be easily it is used to easily detect the face from the image. By analyzing the location of 68 points on the face, the detector can identify facial landmarks including the eyes, nose, mouth, forehead, and eyebrows. Once the ROE selection is done, the image is then processed for color space transformation. So ROI extraction is important as it focuses on the most relevant areas for bilirubin detection, which will be more accurate, reducing the risk of false readings mainly, and it also reduces the variations in lighting by selecting the correct region from the image captured. The next critical step of the skin detection is color space transformation. Color space transformation is a critical step in the process of detecting bilirubin. In bilirubin detection, it is essential to isolate the color variation that correlate with bilirubin levels. Since skin color changes due to bilirubin are subtle, they may be overshadowed by variations in lighting or shadows in the RGB space. All the captured digital images are available in the standard color space RGB. Mainly, this color space transformation is done in order to find the channels which shows the greater level of yellowness or the greater level of bilirubin in the newborns, and it also reduces the effect of lighting, and thus the images which is captured in the RGB color space is thus transformed into another color space. The RGB color space represent images by combining red, green, and blue channels. This RGB color space mainly act as a default color space. Mainly, there are three commonly used color spaces. The first one is YCBCR, and YCBCR, Y represents luminance, CB represents blue difference, and CR represents red difference. Mainly, the CB and CR are representing chrominance. Next is lab. It consists of three components: lightness, A, which represent green to red, and B, that represent blue to yellow. And the third color space is HVS. It separates color information from brightness and saturation. H represent hue channel, which is useful for isolating skin color variations. S represents saturation, and V for value. The importance of color space transformation are it will improve the accuracy, and it also enhances the feature extraction. The first one is RGB color space. The image obtained from the smartphone will have a default RGB color space, which is red, green, and blue channel. The RGB color space is useful in the initial stages of skin color analysis for bilirubin detection. Thus, by analyzing the individual R, G, and B channels, information about the skin color changes due to bilirubin levels can be extracted. For detection of bilirubin, here we are transforming RGB color space to YCBR color space. The YCBCR color space separates chroma from luminance, making it easier to remove the luminance component and reduce false detection. If luminance is not separated from the chrominance, variations in brightness could be misinterpreted, such as mainly changes in skin color, leading to inaccurate detection of jaundice. The B in RGB and CB in YCRCB channels are the primary channels that is used for detecting the bilirubin detection. Mainly, these are the channels which show a greater concentration of yellow color pigmentation. In skin color detection, the skin colored regions and pixels are identified in each channel, and from these main color spaces, RGB and YCB-CR. The RGB representation of the brightness value variations can obtain by using the following equation. Here, this is the given equation where E lambda is the light spectrum and lambda is the wavelength of incident spectrum, and R lambda, G lambda, and B lambda are the sensitivity functions for R, G, and B channels of the color image. Here, R is equal to integral E lambda R lambda into D of lambda, and G equals integral of E lambda G lambda and into D of lambda, and P equal to integral E lambda B lambda into D of lambda. The color representation of YCBCR color space can be obtained from the by using the following equation. An excess level of bilirubin creates a yellow discoloration in the skin. The transformation from RGB to YCBCR color space is done in order to observe the subtle discoloration, that is yellow pigmentation that occurs to the newborn baby. The next step in the skin detection is feature extraction. Feature extraction from the infant's image is done by calculating the mean value of color channel of RGB and YCBCR color spaces. The mean value of the brightness pixel for RGB channels within the selected ROE can be calculated as follows. FR, FG, FB equals sigma x y ROE into ix of x comma y divided by region of interest, where ROE is the size of selected region and I of x comma y is the brightness pixel value at image location x comma y for RGB channels. Therefore, the feature vector is given as f is equal to fr comma fg comma fb comma fy comma fcb comma fcr. These above feature vector are used as 6 features for the detection of bilirubin levels from neonates. Next stage is bilirubin estimation using machine learning algorithms and models. Regression models and multiple methods of machine learning can be used to detect the level of bilirubin from infants' images. A dataset of neonates' images with their bilirubin values are used for training the model. Most commonly used methods for detection are XGBoost algorithm, random forest, and color detection methods using statistical features which utilizes the KNN algorithm. The first algorithm which we are going to discuss is XGBoost algorithm. Gradient boosting algorithm, that is extreme gradient boosting algorithm, is a popular implementation of gradient boosting algorithm. It is mainly designed to be highly efficient, flexible, and portable. The gradient boosting algorithm mainly starts by making a single leaf. The leaf represent an uh, initial guess. The first guess is the average value. Then the gradient boosting builds a tree. This tree is based on the errors from the previous tree. Then it scales the tree and average value is then calculated. Here, the XGBoost is an extreme machine learning algorithm which is used with large complicated data set. The mainly the first step is the initial prediction. Each tree start as a leaf, and then the second step of the method mainly includes building trees. These trees are developed to correct the errors from the previous trees. By correcting the errors of these trees, they will predict a certain value, average value related to the detection or regression analysis. Each tree of the gradient boosting, that extreme gradient boosting algorithm, are trained with certain data sets, and these new trees will get trained with the residuals, and thus the new tree will predict the new residual value. On updating the predictions, they will add the prediction from the new tree to the previous prediction. Also, the weights are after each iteration, weights are updated based on these errors, and thus the higher errors gets higher weights, and thus they will finally predict an value that correlates with the bilirubin. This is the main structure of XGBoost algorithm. As I already mentioned, each tree will learn from the previous tree by correcting the errors that is found in the previous tree, and on updating the new weights, they will predict the new values, and by taking the aggregate of all the prediction, the final prediction will be done. The XGBoost model is mainly trained using the label dataset containing the jaundiced and non-jaundiced images along with the bilirubin level. The feature vector contains the corresponding bilirubin levels for each image. XGBoost model will calculate the feature responsible for the bilirubin levels from R, G, B, and Y, CB, CL colors, channels. The B and CB channels have higher importance, and the values of both channels were compared with the predefined threshold value to classify whether the infant is jaundiced or not. XGBoost algorithm utilizes decision trees and performs well by assigning weights to the features and improving prediction through the ensemble of trees. This is how the XGBoost algorithm predicts the values by correcting the errors and thus predicting the new values from the existing trees. The next machine learning model is random forest. Random forest is a supervised machine learning algorithm which is used for regression and classification problems. RF is developed based on decision tree, and it is a regression algorithm. The feature vector extracted from the images with labels are used as the training data set to the model. Here, as we can see from the image block diagram, first we will obtain the images of the baby, and through the skin detection method, which includes region of interest selection, skin color analysis, that has color space, that perform color space transformation, and then it goes to the histogram for RGB and YCRCB channels, and the results obtained from these histograms are then taken to the random forest algorithm for comparison with the database or the data sets, and then they will display the result which produces or which predicts whether the infant is jaundiced or not. This is the log diagram for RF algorithm. In this algorithm, the training set is mainly divided into subsamples, and each subsample will develop into a tree which makes the prediction. And from the prediction from the each tree is taken, and the average of the or the based on the majority voting, they will decide the final value for the detection. The next method is detection of bilirubin by color detection method. In color detection method, mainly it uses KNN classifier for the detection after performing the pre-processing steps, which include ROI extraction, color space transformation, and feature extraction. The features are taken to the KNN classifier for classifying the infant images based on their condition or the level of bilirubin. This method uses various texture like mean, standard, skewness, kurtosis, entropy, and standard deviation. These statistical features are then fed to the KNN classifier. These are the main texture that act as the features to the algorithm to classify the infant from the jaundice condition. The next is the observation and inferences. XGBoost algorithm has shown greater or higher accuracy in detection the jaundice by estimating the level of bilirubin from the infant's images, and it has shown higher precision than RF model These are the results obtained from the XGBoost algorithm by utilizing XGBoost algorithm. Here, the values of the red, green, blue, Y, CB, and CR channels are obtained, and then the values of the B and CB channels are correlated, and then the condition of the infant is detected whether it is normal or jaundiced. Next is the result of RF algorithm. Here, similarly, the values of all channels, that is red, green, blue, Y, CB, and CR, are obtained. And by comparing the values of B and CB, the condition of the infant is detected. Apart from the XGBoost and RF algorithm, there are several other regression models which can be used to detect the level of bilirubins from the infant's images. That includes KNN linear regression and also support vector machine. But as based on the literature survey, we have found that XGBoost and RF has greater accuracy compared to other techniques. But the XGBoost algorithm outperformed among the all other algorithms, that includes RF, that is random forest, KNN, and also support vector machine. From the table, it can be observed that the XGBoost has shown greater accuracy than compared to KNN classifier and random forest, with a percentage of 98.6% and it has a precision of 99%. So the XGBoost algorithm outperforms among the other algorithms. Next is advantages of using the image processing technique for detecting the level of bilirubin. The first advantage is it's non-invasive. It is non-invasive and pain-free, and it also allows frequent monitoring, and it provides the results very quickly and very efficiently. They provide accurate results, and apart from using the blood test, we can perform image processing techniques, which reduces the risk of infection, and it also improves accuracy with advanced algorithm. As we are using different types of advanced algorithm, the accuracy of the result will increase, and it is also a cost effective method which is compared to other methods, that is transcutaneous bilirubinometer. Moving to the conclusion, this seminar mainly explores the method for detection of neonatal jaundice by image processing technique, analyzing a non-invasive method for estimation of bilirubin from neonates images. Here, we are leveraging a skin color analysis and sophisticated algorithms for detecting the level of bilirubin from the infant's image, which act as a non-invasive approach and also offers a safer, more comfortable alternative to traditional blood tests, allowing for frequent and accurate monitoring of bilirubin levels. And this technology will reduce the risk and also the discomfort that occurs in the newborn babies as well as parents due to the frequent blood draw or blood test, and it also prevents severe complications, and it provides accurate result and also enhances safer environment for the neonates. Next is the future scope. This method can be used for integration with other wearable devices. This helps in personalized monitoring. Even the parents can make use of this image processing technique based detection of bilirubin, and they can monitor even from their home itself, and it can be globally accessible. And this method can also be expanded to other conditions also. These are the references which I took for this seminar. With this, I'm concluding. Thank you.